go ahead and get started. Today it's my great pleasure to introduce Sharlene Santana. Sharlene has come down from University of Washington, where she is an assistant professor. Did her undergraduate in Venezuela. Then she went to University of Massachusetts Amherst, no H, pronounced H, because H is the word, where she did her PhD with Betsy Dumont, working on functional morphology and biomechanical analyses and ecology of bats, so biospheric bats. And then she was awarded a fellowship at UCLA through the Institute for Society and Genetics, where she postdoc for 2 years here. And then she got a position, in fact, a professor position at University of Washington, where she's there now in the second year. And so we're very grateful, happy to have you back in Los Angeles, and looking forward to your talk about functional morphology performance at the university in Max.
Yeah, it's really great to be back and see so many familiar faces. I had a great, awesome experience when I was here at Mike's lab and at the Institute. I think my only regret is that it went too quickly. So yeah, today I'm going to tell you about this research that I've been doing on in different aspects of their diversity. But first I'll start with the general picture of what really drives my interests and what drives the research in my lab. We're very interested in understanding the major patterns of diversity in mammals. In particular, we're interested in understanding what drives the patterns and mechanisms of disparity in morphological and also lineage diversity. So if you look at mammals, you will notice that there are some groups that are overly represented in terms of species, for example, rodents and bats. And in particular, bats are extremely diverse in terms of their function too. So I'm very interested in understanding what's going on at these little parts of the phylogeny where those explosions in morphological and lineage diversity occur. And a lot of the diversification in bats and also in other mammals has to do with feeding. This colonizing new feeding niches has allowed a lot of mammals to diversify and thrive within these. So today, I'm going to focus on understanding then how this different evolution of feeding strategies relate to the diversification within bats. And I'm going to do that by exploring different aspects of ecomorphology, which is basically the field that tries to understand how the evolution of different morphological and also behavioral traits can drive the evolution of differences in performance across species. That is, how the species use their morphology for ecological relevant tasks. And then how that also then enables differences in ecology. And if we study all these factors within a context, we can be able to understand the different patterns in lineage and morphological diversity. So I'm going to focus today on bats because they are an extraordinarily diverse group. As I showed you earlier, they have over 1,000 species, and also they're very diverse in terms of their dietary ecology and in terms of their morphology. And the major question that I'm going to explore is how then this diversity in cranial morphology has evolved in bats, and also then how it maps into functional performance and then so exploring all those different aspects of the morphological paradigm. Here's a little outline of the major aspects I will be talking about today. I'll give you an overview of how cranial morphology evolves in bats, and then how that morphological diversity maps into performance diversity. And later on, what's the ecological significance of all these differences in morphology and also how they pertain to lineage diversification. So I'll start with talking about this evolution of cranial morphology in bats. So in order to understand the evolution of cranial morphology in this group and in mammals in general, we need to go over just a couple of concepts first. And one of them is the concept of modularity. So modularity is a pervasive phenomenon across organisms that explains both the integration within and the autonomy among parts, in this case anatomical parts. So integration is basically the relationships that maintain the proper function and the structure of these parts. So how how they covary with one another. Whereas autonomy is the separation among those parts, or there is the individual variation and evolution among parts. And this enables structures then to adapt in different ways when there are conflicting selective pressures. Now, modularity is a very pervasive phenomenon across the tree of life, from insects to the skulls of many vertebrates. And one pattern that comes up continuously is that when there are higher levels of autonomy, when there are structures that are subdivided into many different parts, this can enable high levels of functional and also so we would expect that whenever we see a group that has lots of morphological diversity, we would see high levels of autonomy or modularity. But that is not the case for mammals. Even though if you look across mammals, we see a lot of morphological diversity in terms of their skulls are actually formed by a very simple modular template. So Goswami in 2006, this is broad phylogenetic comparative study in over 100 species of mammals. And he did this line marking procedures, geometric morphometrics, to estimate the different components, the different modules of the mammal skull. So here's a broad representation of what they found. They found that the mammal cranium is formed by two major modules. One, which is the orofacial region, or the rostrum, and then the other big module is the basic cranium. And also, in theory in mammals, the dentition, the molar dentition, constitutes a separate module. So basically, if you just look at the skull as a whole, there are only two modules here, which is really surprising given the diversity that we see in mammal skulls. Now, these two major modules can be associated with the major functions that these areas have. So the rostrum is mostly implicated in feeding, and the basic cranium is implicated in supporting the brain. But there are many different groups of mammals that have evolved novel functions to their skulls. And one example is bats. So there are several families of bats that have evolved the ability to produce sound through their noses. These are called nasal echolocators. So they produce their sounds in the larynx just like the rest of mammals do, but the sound comes out of their noses. And some of these groups of bats have evolved these chambers in their nasal cavities that basically act as resonating chambers for the echolocation calls. So it's a very distinct type of morphology, and the one that I'm showing you here is the family Rhinolopidae. So this novel function of the skull that is present in bats could have driven the evolution perhaps of a separate module. So we wanted to use these nasal echolocating bats to investigate the question if this novel function of nasal echolocation has reshaped this two modular template of the skull of mammals. And that could lead us to better understand the evolution of morphology, of cranial morphology in bats. So to answer this question, we basically tested two hypotheses conducting geometric morphometric studies. So we tested the hypothesis of the two major modules that are found in all mammals, the rostrum and the brain case. And then a second hypothesis in which this nasal cavity, the nasal chamber, would present a third module in addition to the rostrum and the brain case. We did this across 22 species that presented a lot of variation in terms of the size and shape of the nasal cavity. And we did a geometric morphometric analysis. We adjusted for phylogenetic signal and also for differences in size across species. And we use this RB coefficient to test these two different hypotheses. It's a measure of how well integrated those parts are. So despite the broad variation that we see across the skulls of rhinolopid bats and other nasal echolocating bats, we found that our three-module hypothesis was not supported. In these bats, still, these two modules are still persistent and retained. And that is evident here by this relatively lower RV coefficient, and it's not significant, and higher RV coefficients for the two-module hypothesis in any view that we see the skull. So this was a really surprising result for us because given this new function, we still have these two modules that are really persistent. And we still don't have a good answer of why they have been maintained across the diversity of mammals. One potential explanation is that it has to do with the embryological origins of these two parts of the skull. So the rostrum derives from the neural crest, and the brain case derives from the neurocranium. And even though these two tissues are not integrated in the same way as they are in the skulls of adult mammals throughout ontogeny, it's possible that just basically this subdivision of different tissue origins could be driving this variation. So the question that follows is, okay, we have this relatively simple modular template. Does that restrict somehow the ecological and functional diversity of the skulls of bats or other mammals? And this is a question that I'm going to give you two answers. I'm going to give you the quick answer here for nasal echolocators, and then I'm going to give you the broad answer just throughout the talk as we explore the evolution of cranial diversity in other families. So to answer this question for the nasal echolocators, we did a comparative study of the cranial shapes across a geographic range, which is represented here, so a very broad geographic range. And we matched this with also ecological variables. And what we found is that there's actually, the answer to this question is no. There's a lot of convergent evolution in the shapes of the skulls of bats that inhabit similar regions, which will be correlated to the ecological features of their landscape. So for example, bats that live in Europe and Africa tend to have taller brain cases and shorter rostra, and this could enable them to eat harder prey. Whereas species that live in Asia and Australia tend to have much larger nasal cavities, and this could be correlated with their ability to produce differences in echolocation calls for particular prey types that inhabit in these areas. So we see that despite this two cranial module, which has been two cranial module model that has been maintained over 100 million years of the evolution of mammals, we still can see broad morphological diversity in mammals. And as I'll show you throughout the talk, there's also a lot of functional diversity as well. So that takes us then to mapping then how this diversity in morphologies that we see maps into actually what animals are doing with their anatomy, so their performance. So if we look across the diversity of cranial morphologies in bats, we can describe one major axis of variation that describes basically the whole diversity. Rostrum length is a very important feature. On one extreme, we have species that have long and narrow rostra, and these are skulls that, because they have this long and narrow rostra, they tend to have a low mechanical advantage. That means that they're really bad at translating muscle force into bite force. So these are weak biters. And then on the other extreme, we have species that are the complete opposite. They have really short rostra. And if you look at this skull, and if you've ever seen a primate skull, you will notice that these are snouts that are shorter than most of the primates that you've ever seen. And this enables these species to have a high mechanical advantage, to bite relatively hard for their size. And then we have all the variation that lies in between. So this kind of major trend that I just described, where there's only just one major variable that is mapping into a performance feature, can be described as a one-to-one mapping relationship. When we have one morphological feature, in this case, this relationship of the lever mechanics within the skull, or the mechanical advantage, that maps linearly into a performance rate, which is bite force. So shorter snouts, higher bite force; longer snouts, weaker bite force. But as you might imagine, in nature, things are never this simple. Usually anatomical and functional features are more complex systems. And in that case, they usually conform by many different parts. We have many different morphological or anatomical components that are contributing to one function that then allows a performance rate. And this can get, of course, much more complex. There might be interactions among all the different morphological parts as well. And this can enable a relationship that is called many-to-one mapping. And Mike is very familiar with this because he was really involved in this type of research. And in this type of scenario, there are several different very interesting outcomes. One of them is that each one of these morphological parts is more free to vary or evolve across different species, because selection is acting on the performance feature and the result of those anatomical parts. It's not acting directly on the shape of each one of these different parts. That also means that there can be a lot of morphological variation that does not necessarily translate into functional variation. And this can be a problem if you're trying to understand performance or function on the basis of morphology. If you're using museum specimens to infer functional traits, if you have a many-to-one mapping relationship, you might be in trouble because the relationship is not linear. So this type of relationship has been, oh yeah, I always forget that I have this quote here. This is a good quote from Albert Einstein that I think matches very well this many-to-one mapping relationship, that not everything that can be counted counts. There might be morphological features that really don't mean anything. Okay, so this type of relationships of many-to-one mapping have been extensively described for the feeding apparatus of fish, also for locomotor abilities in lizards. And we have recently done a lot of work on the facial color patterns in primates, and we think that this might also be going on in this type of character. So you can have a pattern that is just as complex in one species and another, basically by using different colors in the face. So this species is basically the negative of that other species, but the number of colors is still the same. So this seems to be a very pervasive phenomenon across different organismal traits. But despite this, this type of relationship has not been investigated in the case of the feeding apparatus of mammals. So that's what I would like to do. I would like to contrast the hypothesis of one-to-one mapping and many-to-one mapping across the whole feeding apparatus of mammals. But today, I'll just tell you the results for bats. Okay, so what are the many parts and many functions of the feeding apparatus of bats and just mammals in general? So in terms of morphology, we have, of course, the skull, and then we have the muscles that close the jaw adductors, so the temporalis, the masseter, the pterygoids, etc. And then we can describe features like the shape of the skull, the levers, the out lever and the in lever. And then for the muscles, we can measure things like the mass of the muscle, fiber lengths, the density of those different fibers, and also how they are arranged within the muscle, the pennation angle. And then we can relate those to different physiological and mechanical functions. So for the skull shape and out levers, we can calculate the mechanical advantage. For the muscles, we can calculate physiological cross-sectional areas and also muscle forces. And then these are two functions that in turn can contribute to performance traits. And those performance traits can be, for example, bite force, and the closing and chewing speed. And these are both characteristics that are important for the ecology of different species, because how hard these animals bite is going to determine what kind of foods they can eat in nature, and also how fast they can eat them will enable them to be better competitors in the case of scarce resources. Okay, so as I showed you earlier, the relationship between skull shape and mechanical advantage is very simple. It's linear, it's a one-to-one mapping relationship. So today I'm going to focus on the myological aspect of this system, that is on the cranial muscles, because this is a very unexplored aspect of this system. So I'll show you now the data that we collected to test these two hypotheses. We did this across 40 bat species from 5 families, and this is a small number when you think about the number of species of bats that there are. There are over 1,300 species of bats. But believe it or not, this is the largest data set that we have of this kind across all of mammals. So this really speaks to the need of this kind of data, quantitative data on muscle morphology, not only in bats, but in other mammals. So if somebody's looking for a project, there you go. Okay, so we collected the following data for all these species. Muscle masses of all the major jaw adductors. We did chemical digestions of the muscles, separated the individual muscle fibers and measured their lengths. And then we had to source from the literature the density of those fibers and muscles. And these are not pinnate muscles, so they have a pennation angle of 0. And then we used those data with standard equations to calculate physiological cross-sectional areas, and also to scale that then to muscle forces. And then we matched those data with performance traits that we measure in the field. So we measured chewing speed versus different species, and I'll just show you this little video here of a bat eating a little cricket. And bats are very eager always to eat whatever you feed them in the field. So they're really great to collecting this data, so we can get data on chewing speed. These videos using the same kind of curve. And then we collect performance data that is bite force with this little gadget that we take to the that allows us to quantify in newtons how hard the different species of bats bite. And here's just a little close-up for how angry bats look when you they're really good also for gathering this data. So the next thing I'm going to show you is what kind of lines of evidence we use to compare these two major hypotheses, one-to-one mapping and many-to-one mapping. And something that I found is that there's an established framework to comparing these two hypotheses. So here's a proposition of things that we could look at. So first, we can look at the relationship among these variables. So if we were to, for example, plot how different the species are in terms of morphology, so morphological disparity, in a one-to-one mapping relationship, we would expect that that would scale linearly and positively with functional and performance disparity. That is, as species are more different in morphology, they're also going to be more different in morphology, in function and performance. Whereas that would not be the case in a many-to-one mapping relationship. We could have evolution of different morphologies, not necessarily translate into disparity in performance and function. The next kind of major aspect that I wanted to look at was the diversity. And you can visualize the spread or the variance across these different axes. So in the case of one-to-one mapping, we would expect that we would have a similar spread, similar diversity in morphology that scales linearly to functional and performance diversity. And that would not be the case for many-to-one mapping. We would have higher morphological diversity than functional and performance diversity. And then finally, if we think of a one-to-one mapping relationship, we're thinking about traits that are evolving in a very linked fashion. So we would expect that the rates and where those change across a tree would be similar. So morphological, functional, and performance traits would have similar rates of evolution that shift around the same times. And that would not be the case in many-to-one mapping. We would have higher rates of evolution for morphological traits. The morphology is changing more than the other traits. All right. So we did this across the species. Of course, we had to take into account phylogenetic relationships, but also sex relationships. This is the smallest species in our data set, that weighs about 5 grams. And this is the largest species in our data set, about 120 grams. So we took into account both of these factors by running phylogenetic generalized least-square regressions. We took residual of all these variables on body mass. And then we used these residuals on the rest of the analysis. We calculated disparity as the Euclidean distance to the centroid in morphological, functional, or performance space, which is a relatively standard way to do it, so how far the different species are from the centroid of the distribution. We calculated diversity at the coefficients of variation in each of those disparity axes. And then we looked at the rates of evolution using the Otero method, which allows us to really nicely map how rates of evolution change across the phylogeny and their direction and their significance. Okay, so I'm going to show you this evolutionary rates first, and then I'll continue with the different relationships. So if we had remember, a one-to-one mapping relationship, we would expect that these rates would be relatively similar. I would like you to focus on these background rates of evolution because the pattern there is a little clearer. So what we see here is, for the most part, these rates are relatively close, except for fiber lengths here, which have a relatively low rate of evolution. And this is an interesting aspect to me because it might be that there's some sort of constraint on the evolution of fiber lengths that we're not aware of. But for the most part, they're relatively similar. They have relatively similar changes in the where the rate shifts, and they're located about the same points in the phylogeny. So this kind of gives some support to that one-to-one mapping relationship. Similarly, we find that morphological disparity here in the x-axis tends to map linearly and positively to functional disparity. And we see that in terms of the coefficients of variation, they're also relatively similar in these two axes here. Now, we run into a problem when we look at mapping functional disparity into performance disparity. There's a flat line. And even though the coefficients of variations are similar, this contradicts the results that I just showed you, and it contradicts the results that I'm about to show you too. So this leads me to believe that there's something wrong with this data set. And what I think might be going on is that these constants that we're sourcing from the literature to calculate PCSAs and muscle forces might not be the right kind of data to use for these bats. Actually, some of these data come from other vertebrates. And we don't know how these different traits, muscle stresses and fiber densities, evolve even across different mammals or within bats. So by introducing just these constants in our equations, we might just be blurring the actual patterns that there exist. So there's a lot of need also for these kinds of data to generate. Okay, so like I said, the reason I believe this is wrong is also because when we look at how morphological disparity maps into performance disparity, we find this like really beautiful positive relationship too. And this is really great news for all of us who have been using morphological proxies of performance, at least when it comes to chewing speed and bite force. They really map very well. And the coefficients of variations are relatively similar too. So this points out to potentially a one-to-one mapping relationship if we look at all these bats as a whole. But I put a question mark there because I wanted to point out something that is interesting too if we look at these data in terms of differences in dietary ecology. So if you look at the green dots here, those are frugivores. And if you were to trace a regression in these two axes, you will see they do have a positive one-to-one relationship. But if you look at the purple triangles here, which are the omnivores, that would be a flat line. So if you were to separate your data by diet, you might get different norms between these two variables. So there might be different ways in which you can be an omnivore. There might be just a relatively simple and easy way and lots of selection on how you can be a frugivore. So this raises an interesting point that when we look at these data, we might also have to take into account the differences in ecology. Okay, so just to summarize then these parts of the results, we found some support for this one-to-one mapping relationship in terms of morphology, in how morphology maps onto function and into performance. And we're still not sure about this relationship until we can generate actual data for the muscle tensions and muscle stresses for bats. We also found some support in terms of the diversity, the spread across these different axes, and also in terms of the rates of evolution. And I also wanted to add the caveat then that these relationships are dependent on ecological effects. Depending on the different guilds that we are exploring, we might have a one-to-one mapping or many-to-one mapping relationship, and we should be aware of that. Okay, so that smoothly leads me to the next step, which is exploring the ecological significance of these different traits. So how this connection between morphology and performance then maps onto the ecology of species. And I'm going to explore these relationships by looking at my most favorite family of bats, which Mike mentioned, the family Phylostomidae, the neotropical leaf-nosed bats. And these are bats that are also nasal echolocators, and they have this big structure here in their noses, the leaf nose, that's why they have their name, which enables them to direct their echolocation calls. It's a relatively large family of bats, over 150 species with great variation in size, and they're distributed from the southern US all the way down to basically the middle of South America. Now, this is a great family to explore these connections between morphology, performance, and ecology because they represent the highest diversity in terms of dietary specializations. This family includes species that are insectivorous, which is the ancestral condition for the group. It includes the only 3 species of vampire bats that exist in the world. It has carnivorous species that eat frogs, lizards, birds, even other bats. And then 2 radiations of bats that rely on plants for their feeding. So nectarivorous bats and also a big radiation of frugivorous species. And associated with this large radiation in diets, we also have a large diversity in terms of cranial morphologies, as you can see here. And these skulls are scaled, so you can also see the size differences. On one extreme, we have those really long snouted species, those are nectarivores. And at the other extreme, we have the really short-faced bats, and these are frugivores, and then everything else in between. So I wanted to then map how is this cranial diversity associated with diet. And the way to establish these connections has to be through a functional trait of the diet that is meaningful for this major function of the skull, which is the production of bite force. And then for that reason, we're not using traditional dietary categories, and instead we're using dietary hardness categories to look at these associations. So we classified the different species of Phylostomids into several categories depending on how hard the diet was. So the vampire bats and the nectarivorous species, they feed on liquid, so they have their own category. And then we can classify the rest of the species into soft, medium, hard, and very hard food eaters. And this last category includes species that have to crack vertebrate bone or eat very hard steaks. And we measure actually the properties, the hardness of these different kinds of prey in the field just to corroborate that they fit into these categories. So for all these dietary categories, we can make different predictions about how the morphology of the skull and of the cranial muscles might have evolved to match these different mechanical needs. And we can do that by looking at other mammals. So for example, we have animals like hyenas that have to deal with cracking open elephant bone. They have to generate very high bite forces at large gapes. And these are species that tend to have a large temporalis muscle. And then on the other hand, we have species that have to bite hard at low gapes, or species that have to rely on relatively soft foods. So these tend to have a large mastodon muscle. So within our dietary hardness categories, we would expect that the species that feed on harder prey would have a larger temporalis contribution to bite force, whereas the species that feed on softer types of foods would have a larger contribution of the masseter. Similarly, we would expect that the species that feed on harder foods would have a higher mechanical advantage to their skull. So to explore these relationships, we decided to build three-dimensional biomechanical models of the skull. And the reason why we do this is because these models actually allows us to see the relative contribution of all these different parts to function. We can run this model and then look at how much torque each one of the muscles produces, and then what aspects of the muscle contribute to producing a higher torque, and so on. To build these models, we did micro CT scans of the skulls of bats, and then we also dissected the major cranial muscles in ways that I described before, separated the muscle fiber bundles. This allows us to calculate PCSAs and muscle forces, which we can all then load into this computer model that basically simulates how the muscles are close in the jaw and how they produce bite force. And we can calculate then the different moments for each one of these muscles, or how much force they contribute. So by running these models across these different species, across the different dietary categories, we were able to describe then how the myology of the feeding apparatus has evolved, or it matches this category. So we found that the species that feed on liquid have a relatively higher contribution of the pterygoid muscles, whereas the species that feed on increasingly harder fruits tend to have a higher contribution of the temporalis muscle. So this kind of solid aspect of the diet really matches those predictions that we can see from other mammals of the relative contribution of the masseter and the temporalis to bite force. And then the liquid feeders are kind of like the oddball here because they have just reduced all the rest of their muscles. So the pterygoids are apparently bigger, but it's just because the other muscles are smaller. And then we also see this really nice match of the low mechanical advantage for the species that feed on softer prey versus the species that feed on harder prey. So this also matches the predictions that we had from before. So this illustrates then how different morphologies really match the ecology, the dietary ecology of the species. But I wanted to take this one step further and also test how these morphologies have evolved and how, and whether and how they have been selected for these different dietary regimes. But because I study bats and bats can live up to 20 years, I can't do selection experiments like you would do in other organisms. So we have to rely on fitting evolutionary models. And this is what we did. We fitted evolutionary models that involved no selections, a Brownian mushroom model of evolution, versus models that involve selection, so Orson-Uhlenberg models, in which you have a parameter that simulates selection, so we can compare the fit of these two different models and model then if there had been selection in different, on these different traits. And I focus on the mechanical advantage of the skull because we know this is related to dietary hardness, and the temporalis moment, so how much force the temporalis contributes to bite force, because this is the largest muscle that contributes to bite force. And then we use different selective regimes, and those were the same of the hardness dietary categories that I explained earlier. So by modeling the evolution then, we're back to exploring the question of whether and how morphology has evolved to match diet. So if there had been no selection across these traits, we would expect that the Brownian motion model of evolution would be the best fit. So there would be no selection for any of these traits. But that is not what we found. We found that actually there's some evidence for selection on mechanical advantage on the skull. So that the species that fit on softer foods and liquid diets tend to have a lower mechanical advantage, and the species that feed on hard foods tend to have a higher mechanical advantage, higher production of bite force, as evidenced by the fit of this model. And similarly, also for the temporalis moment, we have an increasing importance of the temporalis muscle in producing bite force in the species that feed on very hard foods. So we have these two lines of evidence that support the evolution of these cranial traits in relation to diet, both kind of in an ecological context, but also in an evolutionary context. So one aspect that I wanted to mention just really briefly is behavior too. These bats are not static models. They also use different behaviors when they're eating their foods. But we also found that there's a very strong correspondence of their cranial morphologies to the different behaviors that they use to bite on these different foods, and they also adapted to these biting behaviors. So we've done a lot of simulation, computer simulation models using finite element analysis, which is a technique that allows us to see how the different forces act on the skull. And we have found that these really short phase species that feed on very hard foods tend to be very resistant to high bite forces, and also to the twisting that occurs in the skull when they bite on their prey. They tend to bite their prey, as shown here, by biting one side of the jaw with their cheek teeth. So the skulls are not only able to produce these super high bite forces, but they're also resistant to biting behaviors that enable even higher bite forces. So I just wanted to point that out just really briefly, that behaviors are also an important part of this equation. Okay, so now I wanted to tie all these back to the patterns of lineage diversification, which is, I guess, where I started. How these all ecomorphological relationships actually relate to the differences in how many species are in these lineages. So if you are very observant, you will notice that the frugivores here are a big chunk of the radiation of allostomes. We have many more species than the rest. And in fact, we have found that at the base of this frugivorous plate, there's an increase in the rate of diversification, a very strong and significant increase, which is shown here. We found that this group, which are the stenodermatines, that's the name of the subfamily, have much higher speciation rates than non-stenodermatines or other phylostomates in general. So what could be driving this radiation? Well, we have found that this is highly linked to the two major aspects that I described throughout my talk. So that one-to-one mapping relationship that we observe in the frugivores, that is correlation between morphological disparity and performance disparity, and in particular the evolution of these really short phases that had enabled high bite performance, seem to have enabled the colonization of new niches within the species. And we found a significant evidence, as pointed out, by increases in the rates of trophic evolution. So these stenodermatines tend to have much higher rates of trophic evolution than other phylostomates in general. And these rates of evolution, this speed up in the evolution of the dietary ecology of species, moving them towards a more specialized frugivory, and frugivory that is targeted towards very hard. So what we believe happened is that the evolution of these cranial morphologies that enable higher bite forces enabled them to colonize a new dietary niche that was not occupied by any other nocturnal species at that time, and there was the consumption of these very hard foods. And that might have acted as a for the diversification of the family. So we were really excited to find these results because it was kind of like the first line of evidence of an ecological diversification within bats. So with that, I provide you a really broad overview of how these different components of ecomorphology are linked to dietary ecology, and also to ecological diversification. I've shown you how the evolution of differences in cranial morphologies and a little bit of the feeding behaviors enable differences in feeding performance, especially bite-force and chewing speed across different species. And this have allowed different species to then colonize different dietary niches, from insectivory all the way to very specialized frugivory. And in the case of that specialized frugivory, that might have enabled the ecological diversification within at least one family of bats. And with that, I have many people to thank. This is work that require a lot of field work, so lots of people in different field stations, field work, collaborators, including Mike, and also thank you, Mike, for inviting me, and different funding agencies. And with that, I would like to thank you all for your attention.
I'm sure Sharlene would be delighted to...
Yeah, tons of time for questions. Hi, Blair. No problem. You made it.
I have a question about, so the muscle work is great, because you're not needing a lot more information on that. But, so did you see any increase in fiber length in these phyllostomata that are eating the large fruits, that have to bite things at large gates? That's where you expect to.
Yeah. So the fruit eating so yes, but here's why too. Fiber length seems to scale also with body size, and the frugivores tend to also be larger, so there might be a confounding effect there. But that is something interesting to look at. I did find that fiber length is not as variable as you would think across the different diets. Yeah, but yeah, they're still working.
The coronary process, I guess that very last picture, I was thinking the coronary process looks quite short, maybe.
It's shorter, yeah.
Oh, so it's just kind of like, so saber-tooth cats,
Yeah, right.
minimize the stress, so maybe they're solving the problem now.
Yeah, that's another way to do it.
So I'm wondering, do we know, or and if so, how specific are frugivores that to particular fruits? And also the omnivores, what are they doing? They're
Eating fruits, insects, some of the vertebrates too. The frugivores tend to be relatively specific. So you have among all those big eating frugivores, there are a lot of species that are just targeting just a few species of figs. They're highly sympatric, so there's a lot of competition. So they have specialized into particular species of figs. And then you have another group of frugivores that are not targeting figs, but other fruits or other plants too. Yeah.
Question is based on food and that is, that's flawed. And what you've been showing us here is that you have all these continuous variation. You've got bats with small heads, you have bats with middle-sized heads, you have bats with bigger. And they presumably weigh different amounts in terms of the proportions of the body weight and so on. And that will almost certainly change the biomechanical aspects of body.
Yeah.
Because they're going to be more trunk heavy and muscle heavy and all that kind of thing. Has anybody looked at any of the biomechanical connections between, say, the flight properties of these different kinds of bats and morphology that you're talking about?
Yes. Actually, it's work that's not published yet, but so I'll backtrack a little bit. So there are correlations of the wing shapes with habitat, for example. So species that live in cluttered environments tend to have lower aspect ratios, shorter wings and broader wings. Species that forage in open habitats tend to have lower aspect ratios, so long wings.
How about wing loading?
They have higher wing loading and lower wing loading. But I have a colleague who's doing a study about evolution of cranial properties and wing shape to see what might be following what, and there's no conclusive pattern. Because it could be that the evolution of dietary strategies is driving the evolution of wings because the bats need to get to that particular food resource. Whereas it could be the other way around. Wings are just evolving to match different habitats, and then the morphology of the skull is following because those are the kind of foods that they can eat. Yeah. No. in the field. Yeah. Mhm.
And they can take some pictures of bats and flies.
Yeah. Yeah, we have.
Do a lot of interesting stuff.
Yeah, flight biomechanics in bats are really complex because unlike birds, they're basically hands. So they move all their joints when they're flying. So wing shapes, we have these correlations of wing shape with habitat, but we don't really know what they mean because they can deform their wings when they're flying. So it's a changing morphology. But yeah, that's a whole career. Yeah, yeah, but it'd be really interesting. Mm-hmm.
I just have a quick question. So with respect to like ecological response, a large part of that was described as the vegetation and their calls and whatnot. I'm wondering if you know anything about the shape of the phylostomas with respect to their leaf noses, various proportions of length and breadth, and how that overlays onto ecological response.
That's a great question. Actually, I have a grad student who's working on that because we don't have a good answer so far in terms of how different morphologies of the leaf nodes perform and production of echolocation calls. We only have a couple of focus studies on a couple of species, and it seems like the height of the leaf nodes might contribute to detectability in the vertical plane of prey, but that's as far as we go. We don't know anything about. We don't know about how shape evolves, if it's phylogenetic or if it's functional, or how it affects echolocation calls. So yeah.
Is there more specialization in your work relative to like herbivores where evolution is not
In terms of the shape?
Yeah.
It's really variable though. It's really variable. Yeah. Some of the frugivores have this like really crenulated nose leaves too that we really don't know what they're doing. Yeah.
So in the fish world, we always get excited about the sort of the decoupling of different parts of the skull and hypothesis for example. But with this here, really different pattern where you're getting ecological diversification from a pretty simple system.
Yeah.
And I wonder if you had, could you go back and
Definitely.
plotting the morphological diversity and the morphological and the performance relationships?
Oh wait, hold on. I think go here. Let me just go. Oh, close. That one or the?
Yeah.
That's with function though, which is, I think this one is a better one. Hold on, that one with performance.
Yeah.
It's nicer.
So here, what are we looking at? We're looking at the relationship between the disparity of each one of those points is a species.
Yeah. It's a species. Yeah.
So species that.
I think it was later.
No, this one's pretty.
And disparities in distance, Euclidean distance
From the centroid in morphospace or performance space, or yeah.
So how does it correspond to the, that's like a diagram, I mean, you're in the morphospace, you're here and here and here's that. So this one's going to be close, this one's going to be far, but they could be both carnivore and pecivore. So you have blue ones.
Yes. Right. Yeah, they're not. Yeah, exactly. The Euclidean distances were not calculated separately for different diets. Yeah. It's just how far they are from the centroid.
But not the way in which they're far. Not because they're far. Doesn't say anything about what the shape of their skull is. That everybody who lives this far away.
Right.
All it is how far
Yeah.
away.
Yeah. And these are only muscle data, though. They're not skull shape data, which I thought. So I was really surprised by this result because I thought, well, the relationship with the skull is really simple, but the muscles are composed by many different parts, and there's several kinds of muscles too. So I didn't expect that this was going to happen.
We don't have exactly this kind of plot for fishes that I'm aware of, but I would expect a much weaker relationship between mammals with this sort of thing, which is a pretty interesting, if this is true, that there's like a difference in a sort of fundamental way in which mammals are diversifying versus
well, and also specialists versus generalists too. I think that also is playing a lot here, because I think some of these specialists, there just has been such strong selection on them just to, to tie morphology and function to evolve jointly, whereas that might not be the case in generalists, and I think that's why we're seeing this with the omnivores too.
Yeah, so you're struck by the fact that there's a mutual relationship in omnivores.
There's no relationship in the omnivores. It's only 3 points, but
That's usually true is if you have a simple system and selection is acting on that system, you're going to get one response. Predict that response more easily than if you selection would just be able to act on the performance properties in a much more direct way. I don't know, it seems like there's something that if these are general differences between the way that mammals cultivate versus other vertebrates that have more muscular parts.
Or I wouldn't say mammals, but bats, I think. But bats. Yeah. But one thing that we're doing, we're collecting all this biological data for other mammals, because I want to do all this for a much broader taxonomic sample and see how it pans out. If bats are just the weird group, which they could be.
Yeah, it's really interesting.
Maybe Abby, and then
I was wondering also with this graph, later on you talked about diet hardness.
Yeah.
And if you particularly listed it by diet hardness, you can still get that.
Yeah, I think it will hold. I was thinking about that when I was putting this stuff together, because then I talk about that later. I think the omnivores will still kind of fall in that medium category, and then for the frugivores, for the most part, they're on the hard, because these are mostly big eaters. There are only a couple of species that are not. So those two would. Then for the insectivores, then it's a little bit all over the place. Yeah, I think it's fair. Yeah, and then you have the vampire. There's only one vampire species here, which is. It's 1 point, so can't say anything.
I'm going to give a similar preference to Malcolm did. I'm competing with Malcolm for being the most English person in the room. But I have this following question. So, and this is a generic question. It's not just that slide. So each point on that slide represents a species. But the location of that point depends not just on the species, but also on all the other species, which establish the centroid, right? Okay, which means all of the points are dependent on each other.
Yes.
Right?
I guess so.
Okay. Linear regression, however, which is what we've used here, assumes they're all independent.
Yeah.
And clearly they aren't. So, therefore, I don't know how to interpret the graph.
Yeah, that is a good point.
I'm sure that people in the field have thought about it, though.
Yeah, well, we do this kind of analysis all the time of disparity. So I wonder what's the counter. Yeah, because you are right. They are dependent on one another, because they are.
At the same time, it makes perfect sense to do it the way you and evidently everybody in the field does.
Yeah.
So, but clearly there's a glitch that needs to be addressed somehow. I'd like to know what people think.
Yeah.
It's just like looking at the disease, right? Because if you're on the left side, the bottom left side, it's going to be really close to
Yeah. No, but you're right there. The residual
sort of an outlier with respect to morphological or functional.
Yeah, that's a good point.
All right, any other questions for Sharlene? All right, let's thank you.
Thank you.